So in this video we will tell about the complete development of human, okay, from week 1 to week 6. Okay, the now first of all you have to understand, if you understand this thing, this video, the whole embryo will become so easy for you. Okay, so you have to just so focus on these things. Okay, and you in embryo you have to not about the reading, you have to only and only imagination. You have to become to the imagination power of you become so strong that it will help you a lot. Okay, so now first of all we go from each weeks. Okay, now first of all this is in women, when women or female. First of all, there is a menstruation occur. For example, there have a many follicles that is called primary follicles. Okay, from day one. So from day one, the primary follicle development occur. Development of ovarian follicle occur. Okay. S After that, the development of follicle occur that is called oocyte. This is at this time also the menstruation phase. This is also a menstruation phase. During menstruation, the development of primary follicle also occurs. Okay, so the development of ovarian follicles occurs, and this is the oocyte actually. This is the oocyte, and the outer cell become many cell that is called stratified, become stratified. Okay, this is here is it is a simple epithelium, but there is become stratified. Now after this is 1 week, but not our development week. This is before the fertilization. Okay, so we can't say this is the first week. This is the week before the fertilization. First 7 days occur. After that, the this is the outer cells. Okay, and this is the oocyte. Actually, this is the oocyte where the nucleus everything is present. And now this is the outer cells mass, and there is a cave. This cell divide into two, and between them there is a space created that is called antrum. You have to remember this. Okay, now this become developed, and after developing it's become into oocyte, and the antrum is become so large. Okay, after that, when the when this goes in their tracks. Okay, so it goes into ovary, and here in the ovary, at the site of the ovary, it have ovulations occur at the day 14. Okay, this is day 14, so you have to remember this day 14, and also remember that at day 14 LH surge occur. LH hormone becomes so much greater. Okay, and this become due to this the oocyte become detach and ovulation occur. This is the oocyte. Okay, and the remaining cell become remain in the ovaries, and that become goes to the another cell. Now after the oocyte, when ovulation occur, at the when the sperm come, and the sperm come there in the oocyte, for example, this is the oocyte, and you have to remember the cell is not just like a circle. It is, because many students become confused when we goes beyond. Okay, so you have to always in all embryo you have to think that we are studying about the balls. The balls is 4 dimension, not 2 dimension. this is actually this is actually 4 dimensions. 4 dimensions. Okay, 4 dimensions. One is front, back, left, right. Okay, so you have to understand this. Now in the zona pellucida, this is the ball. Okay, and around this the covering of zona pellucida is the covering of the oocyte is called zona pellucida, the outer covering. The outer covering is called zona pellucida. The outer covering of the ball is called zona pellucida. Okay, we have to understand that oocyte is just like a ball. So the outer covering of the ball is called zona pellucida. Okay, this is cover all the ball. Okay, so zona pellucida. After zona pellucida, the inner seal is called oocyte. When the sperm enter, the zona reaction and cortical reaction occur, and the sperm enter to the cell. The sperm nucleus and the oocyte nucleus become union of two nucleus occur. After the union, the cell have divided. Are you see that the this zona pellucida and this have the same size, not change in the size, but both of cell are divided. For example, this is one cell. After some time, this become divided, but the size is remains same. Okay, so just like this cell divided, but their size remains same. Okay, this is also remains same. After when 16 cells, when this divide into 16 cells, for example, this is a ball, and in the ball there is a zona pellucida, and inside there is a cells dead dividing and dividing. When it do, inside the ball there are 16 cell created. When 16 cells, when 16 cell created inside the ball, we call that stage is called morula. Okay, after that the inner 16 cells have arranged into a structure. Okay, that is called early blastocyst. How? They are arranged into a circle and they have a space created inside. Okay, for example, there is a ball, so all the cell goes to the wall, goes to the wall side. Okay, to the wall side, and have created a space, a free space inside the ball. Okay, now after that the cells move on, and the outer cell is become just like a single cell, simple cell. Okay, that is called tropoblast. Here also a hatching occur. How? This is early blastocyst. Or this ball have made up of a zona pellucida. Zona pellucida. Okay, zona pellucida is a thick covering. Okay, is a covering of the ball. So the inner cells, the cells from the inside, they have hatched. They goes out from the ball, and that is called hatching. When these all cell become go out from the zona pellucida, that is called what? That is called simply and simply just like you hatching. Okay, after when hatching, are you see that I have tell that this is the cells that arrange themselves on the wall of the ball. Okay, ball is four dimensions, just like a ball, and they have covered all the walls of the ball. Okay, so now you see this is not a simple layer. This is actually to the all sides. These are covering all the side. For example, this is covering all the things. This is just like a ball. And now see that at some space there is a inner cell mass. There is a mass of cells that is called inner cell mass. And and on the covering of the ball, there is cells that is called tropoblast. Now again I tell to you that after hatching there is also a ball come. There is, for example, in hatching there is a ball come. Okay to you. For example, I tell to you, in hatching, when hatching occur, this is a ball. Okay, and on the wall of the ball there is a tropoblast cell. There is a tropoblast. Okay, tropoblast covering, and inside the ball there is a and inside the ball there this is inside and inside the ball there is a mass. That mass is called inner cell mass. Okay, now at the at the sixth day the implantation began. Are you know implantation is actually, this is the cells. Okay, the cells of the zygote, etc. Okay, the blastocyst. Now in the mother uterus there is a layer. Okay, and when they embedded, they goes inside to the mother uterus. Okay, to embed it, that is called implantation. So implantation began at the day 6. That is called inner cell inner have inner cell mass and anthropoblast. So are when this cell want to go inside the mother uterus. Okay, so this cell have make an invagination. This invagination go inside. After, this is the tropoblast. We have to remember that this is the tropoblast. So this 1 week stage is complete. At 1 week, at first week, there is what occur, the zona reaction occur, that the sperm come, okay, and the morula stage occur, and the blastocyst and the hatching occur, and the implantations occur. Or implantation, actually it is the at day 6 stage, at day 6. So how you can remember this? You have to easy, if you have to remember day 6 as implantation, okay. Here is a 6 p, and p you can write it like this, 6 p and 6 are same like. So implantation occur at the day 6, okay. Now we go to the next. Now at the second week, or this is so simple, so simple. You have to remember all my points, okay. First of all, n- in the second week, what's happening and what's not happening. Many of students confuse, so much confused in the second week, but second week is so easy, so much easy, okay. And the second thing is, this complete video we will understand in the complete detail, okay. This is just an overview, and this overview help you a lot, okay. You don't need any other thing. Now again, come to the second week, okay. In the first week there is implantations occur, and implantation occur, and blastocyst stage. Now in the second week, the thing is just like you have to understand that you are telling, you are teach learning the balls, okay. The ball, this is the ball, okay. Now this ball have the outer covering that is called, that is the outer covering that is called tropoblast. The outer covering is called the tropoblast. Actually, the outer covering is the tropoblast. Now, if the outer covering is the tropoblast, the outer covering is the tropoblast. So what you have to understand, the outer covering has tropoblast. Now this will enter into the uterus of the mother, okay. And after entering, what will happen? Now wait, okay, I again draw you a ball. This is a ball, this is a ball, okay, to you. And now I come here and to tell to you that this ball has an outer covering, this is a ball, this is a ball, this is the outer covering that is called tropoblast. This is the tropoblast, and there is some, this is the outgrowth. This outgrowth is come due to, this outgrowth is for the implantation, for to go inside to the uterus. So now this enter into the uterus, and this has outer covering that is called tropoblast, and it also have the inner cells that is called inner cell mass. This is inner cells, or this is the inner cells mass, okay. Now I will tell you that the inner cells mass in the second week two things occur. One is the, this is the single layer, okay. This is the inner cell mass. The inner cell mass become, for example, divided into two layer. If this is the ball, in between the ball there is a two layer form from the inner cell, one layer and two layer, one layer, one layer and one layer and two layer, one layer and first layer and second layer. These two layers are formed, okay. Now when these two layers are formed, so the space above these two layers are called the space above these two layers. This is the four-dimensional ball. You have to understand. I will tell you in the videos and the complete animation, but you have now to understand. This is the ball, and you have to divide the ball into two spaces. One is above the disc, the bilaminar. This is called bilaminar disc. The inner cell mass convert into bilaminar disc. The space above the ball, the space above the disc is called amniotic amniotic cavity, amniotic space, and the space below the ball is called umbilical vesicle. Okay. Now let's go on how the outer covering is called tropoblast. So, or this is actually the syncytial tropoblast. This is the syncytial tropoblast, the outgrowth of the trophoblast, that is called syncytial trophoblast. So in the syncytial trophoblast there is a space form that is called lacuna, And that space, you have to understand that this is the four dimensional. I again and again say that due to this four dimension many students confused. So this is four dimension from all side, just like a ball enter into the uterus, ball enter into the uterus of the womb, and around that there is a space, there is a syncytial tropoblast, and lacuna appear, a space appear in that is called lacuna. Okay, now go on. The are you know this is the tropoblasts. Okay, this is the tropoblast. The outer covering of the ball is called tropoblast, and the outgrowth is called syncytial tropoblast. Okay, and syncytial tropoblast, a lacuna appear. In the lacuna, the bloods come. The blood island, okay, created. Now, the the tropoblast divided into two. Okay, one is like this is the tropoblast, one is this tropoblast, and extra embryonic mesoderm. This is the extra embryonic mesoderm. Okay, so between actually this tropoblast, for example, this is the outer covering. Okay, this outer covering, this is outer covering. Okay, this outer covering divide into two. So between these two layer, for example, this is the outer covering, this divide into two. So between these two a space created. Okay, a space created, that is called extra embryonic mesoderm, extra embryonic coelom. Actually, between the tropoblast a space created, that space is called extra embryonic coelom. Now don't go complete to this. After that, the this is actually the embryonic disc, that is the bilaminar disc. Okay, the glands, eroded gland, for example in the female uterus there is a glands, like the gland erodes and the blood come from maternal blood, this is the maternal blood. So the maternal blood come to the syncytial tropoblast lacunas. Okay, and this become the nutrient is so much nutrient come to here. Now another thing is the, actually you have to understand the ball, that the ball is present in the inside of the uterus. Okay, this is the ball, and we have divide the ball, we have actually the ball, and the ball we have cut the ball and see it from the side. Okay, here is the bilaminar disc. Okay, this is the tropoblast, and between these there is another tropoblast that is called extra embryonic mesoderm, and between them there is a space that is called uh, that is called actually extra embryonic coelom. Now this is extra embryonic mesoderm. Now another from the X, there is a, I have tell you one is the outer tropoblast, one is the inner tropoblast. Here are when the tropoblast divided, one is the outer tropoblast and the another is the inner tropoblast. Okay, after this going on and the maternal blood created etc. This is the coelom, and now are you know that the outer tropoblast invaginates out, grow to the syncytial tropoblast, and these have called primary villi. When this enter, this is called primary villi. Now you have know that we are talking about the, we are talking about the ball. Okay, we have the ball enter into the uterus. Okay, this is the ball enter into the uterus, and in the, and again and again I say to you, there is a tropoblast, there is a tropoblast. Okay, this is the tropoblast around the ball, and the outgrowth come, that is called syncytial tropoblast from the ball, and there is a space created that is called lacunas. Okay, and a space created that is called lacuna. Okay, and again this is the this is the tropoblast. Okay, this is the syncytial tropoblast, and after that sometime the side, the tropoblast cytotrope, the tropo, this is the tropoblast cytotropoblast, this is syncytial. This tropoblast divided into two layers, one is the outer tropoblast, one is the inner tropoblast. Okay, so you have to again think that this is divided into two layer. If you remove this layer from the ball, the another layer come of the cytotropoblast, that is called inner tropoblast. And when if you remove that layer, the another layer so if you remove, and between, if this is the ball. Okay, if this is the ball, so you have to understand If you divide this ball just like this, if you divide this ball, cut this ball, okay, in 4 dimension, in you have see the ball, divide the ball, the space above the ball is called amnion, okay, this is the amnion, this is the amnion, and the space below is called the umbilical vesicle, okay. Now you have to understand, and between this ball there is a thing that is called the bilaminar disc or embryonic disc, okay. For example, I show to you that this the division layer is called the bilaminar disc. There are two layer bilaminar disc, one is the space above the disc and the space below the disc, okay. This is called the extra embryonic disc, okay. Now another thing, we move on, okay. Now in the third week we don't know, we will not discuss the amniotic cavity, etc. We just remove this ball, this embryonic disc, okay. The that is present between, that divide the ball into two layers, into two things, okay. This, okay, we take this and we will discuss this in the third week, okay. So in the you have to revise the second week. In second week there is a bilaminar disc form, and another thing is the syncytial tropoblast, and the syncytial tropoblast form, and the cytotropoblast divide into outer tropoblast and inner tropoblast, and the lacunas appear, and the trophoblast invaded that form a primary villi, okay. And this is the connecting stalk with the mother uterus, okay. Now we will move on into the third week, that what will happen in the third week, okay. Now the overview of the third layer, okay. In the third layer, what we have just an overview, okay. This third layer is so much important and so much confusing, but you have not to confuse yourself. The first thing is in the third layer that we have, in the second layer we tell about the balls. The first, in the first layer, we have, there is a ball, there is an inner cell mass, outer cell mass, the implantation occur, okay. Ball get inside to the uterus. After the ball get inside to the uterus, there is a sensual trophoblast, the syncytial trophoblast and the cytotrophoblast, the outer trophoblast and the inner trophoblast. We have telling all things about this, okay. Syncytial trophoblast out, then lacuna forms in the second layer, and the primary villi. This all about the second layer, and in the second layer there is a bilaminar disc form, okay. Now in the third layer, or you have to ignore the ball, and between the ball there is a bilaminar disc, okay. Bilaminar disc, and you have to just we will tell about the bilaminar disc, okay. This is the ball, okay. This is the ball, this is your ball, okay. This is the ball, okay. Now in between the ball inside the ball there is a disc, in between the ball there is actually a disc. For example, this is the ball, this is the ball, and inside the ball, this is the ball, okay. And inside the ball there is a disc. That disc is called bilaminar disc. Bilaminar bilaminar disc, okay. This bilaminar disc, okay, between the ball, upper cavity, lower cavity, and this. So we ignore all this ball, we just focus on this bilaminar disc, okay. So the bilaminar disc is just like a plate, okay. Just like a plate between, if you see the ball, cut the ball. So if you cut the ball inside, at one side there is just like one plate, okay. One plate and another plate is above this plate, so two plate, that is called bilaminar. That's why it is called bilaminar disc. So in the third layer, these two plate divide, and these two plate become three plate. For example, one plate, then another plate, this is the another plate above this, another plate come above this, and then after the third plate, two plate is already present, okay. Two plate is already present, but in the third, in the in the third week, the two plate become the three plate. How? So we will tell about. So you have to, in the bilaminar disc, the bilaminar disc, the upper plate is called ectoderm, and the lower plate, for example, there is upper plate and there is a lower plate. The upper plate and the lower plate, the the upper plate is called ectoderm and the inner plate is called endoderm. Now, for example, we have two upper plate. This is the upper plate. Okay, this is the upper plate. Now, in the upper plate, there is a folding. There is a folding occur at one side. At one side, there is a folding occur. At folding or invagination occur. Okay, and when this folding occur, they the actually divided. Wait, for example, I show here. This is the one plate, okay, and at one plate there is folding occur, and after the folding, this space become the cell remove from this space. So it's, for example, this is the plate, upper plate, okay, upper plate, and below this the another plate is present. For example, this is the, I again show to you that this is the plate, one plate, one plate, and above this plate, above this plate, the another plate is present. Above this plate, another plate is present. Okay, and this plate, now in this plate, what happened? This plate is present. Invagination occur at this site. Invagination occur, and after invagination, what will happen? When invagination occur, this is the, this is actually the inside. Okay, this is actually the below plate, and above the plate become, and in the above plate, for example, this is the above plate, the invagination occur. Wait, invagination occur in this plate. What happened? Invagination occur. How? It's, let's wait for a, let's watch it. This is the inner plate, and this is the, and above this plate, another plate is present, and another plane, the invagination occur. The invagination occur. This is the invagination. The invagination occur in this plate. So just like this, the invagination occur. Now at this place, what will happen? At this place, what will happen? At this place, the simple thing is happen that the cell from this place, the cell from this place migrate. In this area, the cell from this place migrate into, just like this, the cell migration occur. The cell from the upper, the ectoderm, goes inside. Okay, so this cell, okay, how? So the next thing after this, the one plate is one, this is one plate, the innermost, this is the innermost plate. Okay, and above this plate is present. But the second I have told that migration occur, come here and see, at the day, at the day 16, at the day 16, what will happen? At the day 16, migration occur. This is the migration of mesenchymal cell. This is the mesenchymal cell, the migration occur. So how it will occur? Now see here, this is the plate, okay, and above the plate there is another plate that is called, that is Actually, wait, we will draw it in the highlighter. Okay, so there is no highlighter of the color which we want. So this is the, no, not showing here. So we will draw it on this. But are you know this cell is migrated. So this cell migrate to the inside between this group to this area. So this cell come here. This cell come here. This cell come here. This cell now, and you have know that the outer this cell is, this is also present. This is also present with inside there is a groove. There is a groove inside. So now this is the below layer. This is the second, the mesenchymal cell that is migrated, and this is the upper ectoderm layer. And also this the most below layer become the most below layer also replaced by this cell. Okay, so this this is the ectoderm. This is the endoderm. This is the mesoderm. Okay, the second is called mesoderm, and this is the endoderm. So in the third week gastrulation occur, and this is the gastrulation process. Okay, ectoderm, mesoderm, endoderm, these three things occur, these three things happen. And another thing, important thing is that this the innermost layer is actually, this is the innermost layer. This is not remain same. Okay, in the third week replaced this become replaced by this cell. So the mesoderm and the endoderm, the mesoderm and endoderm derive from the ectoderm. You have to understand, you have to know that mesoderm and ectoderm derive from this place. Now come back again to this, come back come back to here. Now what will happen in the third layer? The three layer, the gastrulations occur. In the gastrulation, what will happen? First of all, primitive streak, develop primitive streak is a group. Okay, and another, the cell from this area go to inside to the cell, and they create a mesoderm and endoderm, two layers. Okay, and now, so this is the layer, this is from the upper side, this is the amniotic cavity, the ball is cut, and this is the upper plate, this is the and below this is the middle plate, and and again below this is the endoderm. Okay, now just the ball is cut from this side, that we see the primitive streak from another side. Okay, what will happen? For example, this is the ball. Now again we have a we we will tell about the ball. Okay, now come here again. For example, this is the ball. Okay, this is your ball. Okay, inside the ball there is, between them there is a, now here, actually here is a three layer form, that is called gastrulation. one one layer, two layer, three layer. What? Three layer are formed. Three layers are formed. One, two, three. Three layers are formed here. And when we cut the ball into this, when we cut the ball from transverse section, okay, from vertically, so this is the primitive streak. We will see this area. We cut the ball at this area and see this primitive streak from this area. So below primitive streak there is a mesoderm, and below that is called endoderm. And now again, when we see from we see to the top, just like a plate. So this is the plate, this is the plate. Okay, we again see to the plate. Okay, and here, now here is a folding occur. Actually, at the 18th stage the folding occur, folding. at the upper neural plate. The place here is called the neural plate. Okay, to the front of the primitive stage is a neural plate. Okay, and this is the neural plate, and this is the neural plate. Okay, you see to the plate from the upper side, and there is a folding occur from two sides. Folding occur from the, for example, this is the I- actually the folding occur in the plate, not in the whole ball, only in the plate. So if this is the plate, and you fold the plate into this, like you you uh, fold the plate from one side, from both of side, so they become like this, from two side, and from upper and lower side. So just like this, the for example, where I draw this plate, etc., there is no place. Okay, for example, let's wait. Okay, this is the plate. Okay, and you fold it from both of side. When you fold it from both of side, the plate become just like this. Okay, just like this. Okay, you fold it from both side, and another, you also fold it from the caudal and the cranial fold, so it become, so it become like this. The plate become, you folded the plate from the upper and lower side, and from left and right side. Okay, so it become like this, the plate become like this. And in this space is called the neural plate. Actually, this space is called the neural plate. And there is a primitive stick we have already discussed, and this is the outer plate, above plate. Another is the mesoderm, and below that is the endoderm. Okay, now, so the from this area the brain develop, and from this area the neural groove develop, and from this area the somite develop. Okay, and this is the neural groove, first pair of somite and the primitive streak, and this is the connecting stalk. So in the detail we will discuss in the later of video. Okay, thanks for watching.